All, my dear first year BSc students, I welcome you all to the online class on zoology. So, in this semester, you have to start with the principles of animal classification, since this is the very basic need for a zoologist to start the study of the animal science. So we have to first learn how to classify the animal, what are the basis for classification, how to classify the animals, how to categorize them, what are the criteria involved, etc. So along with that, we have to go through the system of binomial nomenclature, Linnan hierarchy, and then the concept of species, the criteria for classification, etc. So we are going to study all these in this first chapter of first semester zoology. So now, whenever we strike on the name classification, taxonomy, or systematics, what does actually it means? See, classification is a simple English term which is used to define the categorization of something based on some criteria, right? Say for an example, we classify our students as students belonging to the first semester, third semester, fifth semester. In that, we again categorize them based on their streams, whether they belong to the arts stream or science stream or commerce stream or administration stream, whatever it may be, right? So we try to group the people according to the similarities they possess. So exactly similar thing happens with the animal system also, where we categorize animals based on some similarities they have in common. So systematics means it's the scientific study of kinds and diversity of organisms and of any and all relationships among them. Okay, we are going to study about their diversity, about their types and kinds, and also the relationships among them. There may be some relationships or many relationships, whatever it may be. So all kinds of such relationships of animals will be studied here. So whether it may be phylogenetic relationship, that means the evolutionary history of the animal, or relationships in their anatomy, relationships in their morphology, relationships in their physiology, whatever it may be. Everything is taken into consideration, and then based on the common things they have, they are categorized or grouped into different clades. So this is actually the classification of animals. Okay, so if you look into the history of the classification of animals or living organisms, so since time immemorial, different people have tried very hard to categorize plants and animals or other living creatures just to make their study easy. See, it has been estimated that around 1.2 billion, that means 120 crore animals are living on this planet today. It is just an estimation, there may be more also. Many of them, so about 90% of them have not been recognized, not been discovered, not even been named. So if you are a zoologist or a botanist or a student of biology, you will find it very difficult to study all these 120 crore animals individually, right? So that's a impossible task. So what to do now? Now we have to classify them, categorize them based on their similarity so that you can study them as a group. When you study them as a group, let us say for instance, if you study about vertebrates, vertebrata is a group of animals having the vertebral column. So if you study all the salient features of a group called vertebrata, then by and large you will understand the basic concepts of life forms belonging to the group vertebrata. So you need not to study each and every vertebrate. You can just focus on a single group. So this is not a simple task. Okay, categorization requires lot of effort and this effort of classifying animals has come from time immemorial. Okay, so the very oldest possible evidence of classification today available for us is our own Indian saint Parashara, who has written a book called Vrikshayurveda, wherein he has put lot of efforts to categorize medicinal plants based on their characters, based on their availability, based on their habitat, based on their medicinal uses, etc. Okay. Later on, certain Western thinkers, Western philosophers like Aristotle, for an example, is also called as father of biology. He started to scientifically classify animals based on certain features, certain common characters that they possess. One character he found was the animals which lay egg and the animals which give birth to young ones. He called those animals which give birth to young ones as viviparous, and those animals which lay eggs are called oviparous. This was a crude classification, all animals which lay egg were kept in one group and all other animals which give birth to young ones were kept in other group. But this was an attempt to classify animals. Along with that, he also classified animals as animals with red blood and animals with white blood. Okay. So this was just an attempt to classify animals based on certain similarities they had within them. Okay. Later on, his Aristotle's student Theophrastus, who is also known as the father of botany, he tried to classify the plants based on their height and other certain characters which he seemed to be very important for classification. He classified the plant kingdom into four categories: the trees, which are taller and larger in size; shrubs, which are bushy in nature; undershrubs, they are small plants but are bushy in nature; and herbs, which are not even bushy and also they are small plants. So this is a very simple classification. You can cite as many as examples for this. As well as this was a crude classification, just focusing on one character, there was a height of the plant, right? Trees, shrubs, undershrubs, and herbs. So later on, many efforts were put forth to classify animals based on their different characters, different types of characters. Each scientist went with his own way. Okay, somebody took height as a character, somebody took anatomy as a character, somebody took the different characters. For an example, the presence of beak, absence of beak, presence of fins, absence of fins, presence of wings, absence of wings. Okay, based on their habitat, for an example, the aquatic, the aerial, the terrestrial, whatever it may be. So like that, till the 16th or 17th century, very crude way of classification of animals and plants took place. Okay, each and every scientist gave his own hypothesis, gave his own theory of classification, and classified animals with different you know, principles. But it was Carl Linnaeus, also known as Carolus Linnaeus, who is now called as father of taxonomy, who actually started the biological way of classification. We call this type of classification or taxonomy as natural classification today. He wrote a book called Systema Nature. Along with it, he has written many other books like Genera Plantarum, Species Plantarum, etc. But one of the greatest contribution of Carl Linnaeus is the introduction of binomial system of nomenclature. Before binomial system, there was something called polynomial system, where each and every organism was named with many names. So from 5 or 6 names were involved in that, which was very, very difficult to remember, as well as name, as well as to write, as well as to, fix a particular name to a particular organism. But it was Carl Linnaeus who introduced a system called binomial nomenclature, where each and every living organism is given a scientific name which is unchangeable, which is universal. That means the entire world should accept that, and the same name must be used by all scientists, all biologists, including common people all over the world. So this system made the classification as well as scientific naming of an organism quite easy and bit more scientific and more specific. So because of these contributions, Carl Linnaeus was considered as the father of taxonomy. So whenever we study about Carl Linnaeus, we will be introduced to something called Linnaean hierarchy, where we can find many categories of classification. So let us not worry about the life domain as well as life and domain here. Let us start with the kingdom part here, okay? So Linnaean hierarchy includes kingdom, which is a very big category of classification, and gradually the classification norms narrows, and finally it ends up in the genus and species, which is very specific to a particular organism. Okay. So initially the Linnaean hierarchy starts with the kingdom. Kingdom, we have got 5 kingdom based on Whitaker's classification. Kingdom Monera, Kingdom Protista, Kingdom Mycota, which includes all fungus, Kingdom Plantalia, which includes all plants, Kingdom Animalia, which includes all animals. Followed by phylum. In plants it is called as divisions sometimes. Okay. Phylum is a category that comes under the kingdom domain. Okay. And phylum is further classified into classes, and classes are classified further into orders. Each order is further classified into families. Each families are further classified into genus. Each genus is further classified into species. So these are the 7 categories of Linnaean hierarchy. Starts with the kingdom, followed by phylum, class, order, family, genus and species. So now each category, when you go through the Linnaean hierarchy, from each category the characters or the principles for classification goes on narrowing, and it becomes more and more specific when you go below the Linnaean hierarchy. Okay. So this is how you can, this is a pictorial representation of Linnaean hierarchy. See, we can see the animal kingdom here. Okay. The animal kingdom means all animals. which are heterotrophic, possessing locomotory organs and more. Okay, where you can see many animals put together. It's a grizzly bear, it's a black bear, giant panda, red fox, squirrels, coral snake, sea star, everything came under a single group called Animalia. And then the next character for consideration is presence or absence of notochord. So the sea star, the starfish don't possess notochord, so this was excluded, one became less. Go to the next one, the class Mammalia, were all animals bearing mammary glands. Okay, the mammary glands are the organs which secrete a nutritious fluid called milk for the nourishment of young ones, right? So here snake is now excluded because snake do not possess mammary gland, it is a reptile. Okay, the next class or group of classification is carnivore, all carnivorous animals. Okay, bear, tiger, lion, panther, you know, mm, dogs, cats, everything comes under this group carnivore, all carnivorous animals. So now we have to remove squirrel from here because squirrel is a rodent. Okay, the animal belonging to the rat group, not the carnivore group. So this is excluded now. Go little further, family Ursidae. Ursidae includes all bears, all types of bears. Okay, Ursidae is a family which includes bears. So we have to remove the fox here. Fox is a carnivore but not a ursid member. It's not an animal belonging to the bear group. It is the animal belonging to the dog group. Okay. So then genus Ursus. So red panda is now excluded because this is not an animal belonging to the genus Ursus. And then the last one, species category, only one animal is left out, that is Ursus arctos, because the grizzly bear is the animal having this scientific name Ursus arctos. Hence, this black bear is excluded. Now you can understand how gradually when you go along the linear hierarchy from kingdom to species, the system is becoming more and more specific. So system is now giving more and more specific classification of an organism, right? So this is the best wonderful advantage of Linnaean hierarchy. So again, this is a example of Linnaean hierarchy which is written in the reverse format. Okay, using one species, that is Panthera pardus, leopard. Okay. See, when the system of naming an organism with two names, binomial nomenclature, was initiated, started long ago in 16th century or 17th century. So again, there were so many confusions, so many problems with that, because it is not so easy to bring the entire world into a common umbrella of naming, right? Say for an example, the problem is the common names. So common names differ from place to place, region to region, state to state, country to country, right? The language differs, so the naming differs. In our own state, for an example, let us say Kannada, we have more than 10 names for a single species called dog. And along with that, we have many more names which are, taken from our neighboring languages, Tamil, Telugu, or Marathi, or Hindi, or Sanskrita, whatever it may be, right? So dog alone has more than 25 names in Indian regional languages. Now this creates lot of confusions, because the man who is living in Uttar Pradesh or New Delhi don't know what nai is. And people who are living in the southern India may not be knowing what kutta is. So this is the problem of having a common name for all animals. So region to region the name may differ. At the same time, same animals in different region may have same names or different names, and different animals in different region may have same names. So in order to get rid of all these kinds of confusions, International Congress of Zoology was you know organized in 1898, and they commissioned a organization called International Commission on Zoological Nomenclature, which today formulates some rules and regulations which all the taxonomists who is now ready to discover a species, or who is who has already discovered a species and who want to name that particular species, they have to abide by the rules and regulations laid down by International Commission on Zoological Nomenclature. So what are those rules? Now before going to rules, we have to focus on the principles for classifications, right? So before you name a particular organism, before you classify a particular organism, we have to initially classify them, right? Then we can name them. So what are the criteria for such classification? So one of the very basic criteria utilized by the taxonomists to classify animals is grades of organization. Cellular grade of organization, we have four kinds, right? Cellular grade of organization, tissue grade of organization, organ grade of organization, and organ system grade of organization. So cellular grade of organization is largely observed in almost all protozoans, where a single cell is responsible for each and every physiological and other activities of the organism. It may be respiration, it may be food intake, it may be food digestion, it may be excretion, it may be water intake, expulsion of water, reproduction. Okay, everything movement is taken care by a single cell. Such a kind of organization is called cellular grade of organization. Almost all unicellular organisms do possess cellular grade of organization. Moving further, what is tissue grade? So tissue is a group of cells. Okay, it's a group of cells performing a common function, similar function. Say for an example, epithelial cells. Epithelial cell is a cell which perform a particular function. That's why all organs do have epithelial cells as their covering. Along with that, we do have many other types of epithelial cells. Okay, you would have already studied them. I am not going to give details about that right now. But let us say muscle cells for an example. The function of muscles is to contract and relax so that the movement takes place. So there are different types of muscle cells. Each of them, when grouped together, perform a similar function. Hence, they are called tissue. And tissue level of organization is observed in many sponges, in almost all sponges. In coelentrates, sponges do have cellular grade of organization. In coelentrates, that means jellyfish, hydra, lobelia, all these possess tissue grade of organization, where a group of cells come together, perform a similar function. And what's an organ now? Organ is a group of tissue. Different tissues come together performing a single function. Okay, let us say liver. Liver has many cells. So different different types of cells have come together and they perform a similar function, right? That is called organ system, which is initiated from platyhelminthes. You can see different organs in platyhelminthes, but not exact organ system. Organ system starts with Aschelminthes and annelids. Okay, organ system means different types of organs, they come together, they form a particular system responsible for a particular function. Let us say digestive system, it's a combination of different organs, right? We have buccal cavity, we have esophagus, we have stomach, we have intestine, we have digestive glands such as pancreas and liver. All of them come together and perform a single function, that's digestion, right? So this is organ system level of organization which is observed in higher organisms, higher animals. So after that we have to focus on the symmetry of the organism. So we have got three kinds of symmetry here. The first one is asymmetry. So complete absence of symmetry. If you have seen sponges, you could appreciate the asymmetrical uh, shape of those organisms. If you have not observed, not a problem. So when you come to the college, we will show many specimens of sponges. There's Sycon, Ascon, Leucosolinea, Spongilla, all of them. They are just like heap of cells. There is no specific pattern of their growth. So that is asymmetry. There is no symmetry. Then we have got two different kinds of symmetry. One is radial symmetry and the other one is bilateral symmetry. This picture also you can appreciate different types of symmetry. Sometimes, the absence of symmetry is also called no symmetry, asymmetry, no symmetry, whatever it may be. Then the radial symmetry. See, radial symmetry means all coelentrates do have radial symmetry. See, here is a cnidarian for an example, Metridium, C. anemone. So when you cut this animal in a longitudinal way, that means from the anterior end, this is anterior end, to the posterior end, okay, passing through the central axis of the animal. You have to cut it from head to tail, okay, from anterior position to the posterior position, passing through the central axis, okay. You will get two two body parts which are mirror images of each other. The left half is exactly similar to the right half. If there is two tentacles in the left side, then on the right side there must be two tentacles, okay. This is called radial symmetry. Now how this becomes radial? See, if you are able to cut this particular organisms in any plane, okay, right plane, left plane, whatever the plane you wish to cut it, these animals will be generally circular in nature, okay. So whatever the plane you try to cut it, you will get two equal halves, right? Right half will be exactly equivalent to left half. But there is a second type of symmetry called bilateral symmetry. That means here also you can cut the animal from its anterior end to the posterior end, passing through the central axis. This is very important. You have to pass it through the central axis. But at only one plane you can get two pieces which are the mirror images of each other. Okay, you cannot cut it in this way, you cannot cut it in this way, you cannot cut it in this way. Only a particular pattern of cut, that is from anterior end to the posterior end passing through the mid axis, you can get the exact mirror images, okay, two pieces which are exactly the mirror images of each other. So this kind of a symmetry is called bilateral symmetry. So all the organisms which has got this bilateral symmetry belong to the group called Bilateria, and all the animals, best examples are coelentrates and cnidarians, they possess radial symmetry, hence they are categorized under a group called Radiata. So then the presence of body layers. So these are generally, not generally, scientifically termed as germinal layers. So if you have studied the uh, pattern of development in different organisms in your previous classes, you would appreciate there is a stage called gastrula. If you remember gastrula of frog, okay, this will be something like this with the yolk plug and all. So in this phase of gastrula, actually the bilateral symmetry is established. Along with that, there may be 3 germinal layers or 2 germinal layers. Okay. So the outermost layer is called ectoderm, the innermost layer is called endoderm, and the middle layer is called mesoderm. If any animal possesses all these 3 layers during their gastrula stage, during their embryonic stage, ectoderm, endoderm, mesoderm, if all these 3 germinal layers are present, then such animals are called triploblastic animals. Tri means 3, blastic means the three layers, okay. Triploblastic animals. If the animals do not possess all the 3 layers, say for an example in coelentrates the mesoderm is missing. Instead of that, the mesoderm uh, is missing in these coelentrates, and a space is created because of the absence of mesoderm. Hence, a fluid will be filled in between the endoderm and ectoderm, and that fluid is called mesoglia. So, all animals which belong to this group are called diploblastica, and all animals which have all the 3 germinal layers are called triploblastica. Okay. So once this blasticity is established, that means the body layers are established, we come across one more term called coelom, which is very important for the classification of higher animals. See, coelom is sometimes wrongly termed as a body cavity. Coelom is not just a body cavity, okay. Coelom is something different. Now, what is that different coelom? See, coelom means that is a space created by the bifurcation of or splitting up of mesoderm. Okay. So during the time of embryonic development, the mesoderm splits into 2 pieces, 2 layers. The outer layer goes with the ectoderm, the inner layer goes with the endoderm, and now because of the splitting there is a space created, and that space is called coelom. You can see this space here, right? So here you can see 3 layers. The outermost layer is ectoderm, the innermost, the white colored layer is endoderm, okay, and between the maroon colored one is the mesoderm. So now mesoderm splits into 2, forms a cavity in between, hence it is called coelom. And if the coelom is absent, such animals are called acoelomate animals, absence of coelom, complete absence of coelom. If the coelom is present, then those animals are called eucoelomate. Eu means true. So true coelomate animals. So in certain other categories of animals, for an example, nematodes, the coelom is bit different from true coelom. Okay. Here, the coelom is not completely covered by a peritoneal layer. So peritoneal layer means the layer formed by the splitting up of the mesoderm, right? So that space is not completely covered by the peritoneal layer, and such coelom is called pseudocoelom. So coelom is coelom like structure is present, but that is not the true coelom. True coelom means that is the space which is formed due to the splitting up of or bifurcation of mesoderm. Okay. Next we have something called metamerism. Metamerism is segmentation of the body. So certain animals are metamerically segmented, means the entire body of that organism. Example, all annelids, okay, maybe earthworm, leech, sebella, arenicola, whatever it may be. They have this kind of you know metameric segments in their body, right? You can see here one segment. This is a cut section of earthworm. So first segment, second segment, third segment, like that you can observe many segments. Okay, that is called metamerism. And metamerism is also a best criteria for classification, because true metamerism is observed in many higher organisms, where you can see in almost all organisms above arthropoda, mollusca, echinoderm, anyhow echinoderm, the larval stage only possess metamerism. Some sort of metamerism is present. In adult stage you cannot observe true metamerism. But in all vertebrates you can find metamerism, where the body is categorically divided into 3 parts or 4 parts, whatever it may be: head, thorax and abdomen, right? Okay. So this is how you can see earthworm segmented. And there is one more organism which has got false segmentation. What is the difference between false segmentation and true segmentation? See, in annelids you can see true segmentation, where the segments are present, but each segment goes on increasing in size, as well as they go on growing in size. There will be growth. But in false segmentation, the segments will be added to the head region. Okay, head will give out the segments. The last segments of the animal will be oldest, and the first segment will be the youngest. That type of categorization is not there in the animals which do possess true segmentation. Okay, so this is the false segmentation can be observed in tapeworms.